I am Dan Cleary, and I'm going to be discussing our chapter, The Anatomy and Physiology of Pain, by Daniel Cleary and Mary Heinricher. We have 4 objectives for this chapter. The foundation of this discussion is the difference between nociception and pain. From there, the discussion will include ascending pathways, key nervous system sites, and descending modulation. Before discussing the neural pathways, it is important to distinguish nociception from pain. As defined by the International Association for the Study of Pain, pain is a subjective experience that may or may not be related to tissue damage. Conversely, nociception is the objective neural encoding of noxious or damaging inputs. This chapter will focus on nociception and nociceptive pathways rather than pain. Peripheral nerves relaying nociceptive signals synapse on second-order neurons in the dorsal horn of the spinal cord. These second-order neurons then send projections across the midline to the anterolateral quadrant, where they ascend in the spinothalamic tract. The majority of spinothalamic tract projections go to the thalamus. The spinothalamic tract relays ascending nociceptive signals to the brainstem and the midbrain, but most of the connections go to the thalamus. The nociceptive nuclei of the thalamus can be roughly divided into medial and lateral thalamic nuclei. Medial sites project to areas involved in affective or emotional elements of nociception, such as the medial prefrontal cortex and the anterior cingulate cortex. Conversely, the lateral thalamic nuclei project to areas involved in sensory discriminative elements of nociception, such as primary and secondary somatosensory cortex and the insula. Please note that this division of medial and lateral nociceptive pathways is not as clear-cut as once thought, and we now know that significant interconnections exist between areas. The central nervous system contains an intrinsic system for nociceptive autoregulation, which can increase or decrease nociceptive input to influence behavioral priorities. Forebrain sites, such as the medial prefrontal cortex and the anterior cingulate, project to the midbrain periaqueductal gray, or the PAG. Neurons in the PAG then send projections to the rostral ventral medial medulla, also known as the RVM, whose cells project to the spinal cord. The RVM contains 2 populations of neurons that can influence nociception through descending projections, the on cells and the off cells. Both classes of cells send projections to the dorsal horn of the spinal cord, where they can influence nociception and behavior by regulating activity of dorsal horn neurons. On cells facilitate nociception and produce hyperalgesia, a state of increased perception of pain, such as occurs during acute sickness. Conversely, off cells inhibit nociception and produce analgesia, such as with acute stresses where action must be prioritized over pain. Pain is a uniquely subjective experience, and so we refer to nociception when discussing neural processing of noxious inputs. The transmission of nociceptive signals involves several important relays, starting with the peripheral nerve and then going to the dorsal horn, through the spinothalamic tract, to various sites in both the medial and lateral thalamus, and to multiple sites within the cortex. Recurrent connections via the midbrain and the brainstem actively regulate nociceptive input through action at the dorsal horn. For more detailed information, please refer to the text of this chapter.